tea, the milkman, the paper boy, the evening TV. Everywhere you look, there's a heart, a hand to hold on to. Everywhere you look, there's a face, somebody who needs you. Everywhere you look, when you're lost out there and you're all alone, a light is waiting to carry you home. Everywhere you look, everywhere you look.
Alright, so this is DNA replication. Right here, this giant pink thing is helicase. What this does is it goes through and separates out the two strands of DNA and creates a replication fork. And right here we have the single-stranded DNA binding proteins, and they go along and hold the leading strand still. And then this DNA strand, the leading strand, creates new DNA from the 5' to 3' end. And this is continuously going on. And then down here we have the lagging strand, which has to go from the 5' to the 3' section. And so what happens is we have, I don't even remember what this is called. We have RNA primase, which comes from the replication fork and creates RNA primers, which then primase comes down and goes from the RNA primer and creates new DNA to meet with the lagging strand, the 5' end that it started at.
Topoisomerase is located in the nucleus.
We have type 1A topoisomerase on the left, and then topoisomerase type 1B on the right. Each side you can see has 4 core domains, and each one is depicted through a different color. Now on the left you see it's a lot more straggly and not the same buildup as on the right, and that's why we'll be focused on type 1B throughout the rest of this process. And type 1B is also found in eukaryotes, which is more applicable to us. Now as you see, type 1B has 20 angstrom middle core, which the other one does not have.
Type 1B is a multimeric protein with 4 different domains. It has a 4 crisscross beta strands surrounding by 4 alpha helixes that are arranged in a Rosman fold, which a Rosman fold is a protein structural moffitt found in proteins that bind nucleotides.
On the board in black is the active site of topoisomerase, and in green is the DNA. What topoisomerase does is the enzyme first wraps around DNA and creates a single 3-phosphotyrosine intermediate. The 5' end is then free to rotate, twisting it about the other strand of DNA to relax DNA until topoisomerase religates the broken strands. So this just represents the DNA being cut and being moved around. Take one. Oh.
So this is the process of DNA replication. Without topoisomerase, it would result in supercoiling.
Yep.
The importance of topoisomerase is displayed on the board. The left strand is when topoisomerase is functioning properly, and then an inhibitor is put into place, which prohibits the topoisomerase from working properly, which breaks the DNA and results in cell death. This is used to kill cancer and bacteria cells.
To prevent supercoiling, topoisomerase 1 cuts one single-strand DNA. As it cuts the DNA, it pulls the DNA through the supercoil to relieve torsional strain!